Welcome to the deep dive. Ever wondered, how we inherit our traits, like eye color or height?
It's a fundamental question, really.
Exactly. So today, we're diving into the fascinating world of genetics and inheritance. We're going to explore how characteristics actually get passed down through generations.
And we'll be using Genetics by Mr. Chi.pdf as our guide for this.
That's right. We'll unpack the key concepts from that source.
Yeah, our mission, if you like, is to pull out the core ideas of heredity from this resource. We want to give you a clear, engaging understanding of the basics, the fundamental mechanisms.
Without getting totally bogged down in, super technical jargon.
Exactly. We're aiming for what's most important, most interesting about how we become who we are, genetically speaking.
So what kind of ground will we cover?
Well, we'll start with the basics, genes, alleles, and move up to more complex things like how sex influences inheritance, and even touch on things like biotechnology.
Okay, sounds great. Let's start right at the beginning then. What exactly is heredity, and how does genetics fit into that picture?
Right, so heredity. Well, basically, it's just the passing of characteristics from parents to their offspring.
Like you said, eye color maybe?
Exactly, or height, or hair color. And genetics is a science that studies this whole process.
Yeah.
It investigates how these heritable traits, the physical stuff, even predispositions sometimes, are actually transmitted across generations.
Okay, so when we talk about a specific trait, say a plant height, tall or short or flower color, how do genes and these things called alleles come into it?
Good question. So traits are the things we see, the observable characteristics. Genes are the specific instructions found on segments of DNA on our chromosomes that code for a particular trait.
Instructions.
Yeah.
Gotcha.
Now, a single gene can actually have different versions, these alternative versions. We call them alleles.
Alleles, okay.
And these alleles sit at the same specific spot called a locus on homologous chromosomes. Those are the matching pairs of chromosomes we get, one from mom, one from dad.
So for height.
Right? For plant height, the gene might have one allele for tall, maybe we use a capital T, and another allele for short, maybe a lowercase t. Same idea for flower color. Different alleles for different colors.
And that location, the locus, is important.
Crucial. It ensures that when parents reproduce, they each pass on one allele for that trait at the same corresponding position to their child.
Okay, so we inherit these alleles. How do they then decide what we actually see? The organism's phenotype. And what's the difference between genotype and phenotype?
Right. Your genotype is your specific genetic combo for a trait, the actual alleles you have. So for our pea plant, its genotype could be TT, TT, or TT.
The actual allele pairing.
Exactly. The phenotype, though, is what you see, the physical expression. So for pea plants, both TT and TT genotypes make the plant look tall. That's the tall phenotype.
Why is TT tall?
Because the tall allele, T, is dominant. It masks the effect of the short allele,
Yeah.
T. Only if the plant gets two short alleles, genotype TT, will it actually be short show with the short phenotype.
Dominant and recessive. Those are really key concepts, aren't they? You give us another example just to make it super clear.
Absolutely. Let's think about tongue rolling. Whether you can curl your tongue up.
Yeah, I can do that.
Okay. So let's say there's a gene for that. The allele for being able to roll your tongue, let's call it R, is dominant. The allele for not being able to, little r, is recessive.
Okay.
So if your genotype is RR or RR, you can roll your tongue. That's the dominant phenotype. Only someone with the RR genotype, two recessive alleles, can't roll their tongue.
So R still means you can roll it.
Right, because R is dominant. And we use terms for this too. If you have two identical alleles like RR or RR, we say you're homozygous for that trait. If you have two different alleles like RR, you're heterozygous.
Homozygous means same, heterozygous means different. Got it.
And this simple dominance thing explains how traits can seem to skip a generation and then pop back up later. Puzzled people for ages before Mendel figured it out.
That really helps clarify. Now, the text mentions monohybrid and dihybrid crosses. What's the basic difference there?
Yeah, good point. A monohybrid cross is when we're tracking just one trait, like only flower color or just seed shape. We're following how the alleles for that single gene get passed down.
Mono meaning one.
Exactly. A dihybrid cross, well, di means two. So there we're looking at the inheritance of two different traits at the same time, like flower color and seed shape, seeing how the alleles for both those genes travel together or don't.
Okay, one trait versus two traits simultaneously. Makes sense. Now, the source also points out that dominance isn't always quite so simple. We have complete dominance, like the tall, short plants, but also incomplete dominance and codominance. How do those work?
Right, it gets more nuanced. Complete dominance is what we discussed.
Yeah.
Teat looks just like TT, the dominant allele, totally hides the recessive one.
Okay.
Incomplete dominance is different. Here, the heterozygote, the one with two different alleles, shows a phenotype that's kind of a blend of the two parents. It's intermediate.
A blend, like mixing paint.
Sort of. The classic example is snapdragon flowers. Cross a red one, RR, with a white one, WW. The offspring, which are all RW, aren't red or white, they're pink. Neither allele fully dominates, so you get this in-between look.
Ah, pink flowers from red and white. Interesting. And how is codominance different from that blending?
In codominance, both alleles are expressed fully and distinctly at the same time. Instead of blending, you see both traits.
Both show up. How?
Think about certain cattle breeds. Cross a red-coated cow, RR, with a white-coated one, WW. The offspring, RW, won't be pinkish. They'll have patches of red hair and patches of white hair. Both red and white show up side by side.
Ah, okay. So not blended like pink, but like polka dots almost. Both colors there.
Exactly. Both alleles making their presence known equally.
So, quick recap. Complete dominance, one hides the other. Incomplete, it's a blend. Codominance, both show up separately. That's useful. The source also mentions multiple alleles using the ABO blood group system. How does that work?
Right, so multiple alleles just means that for some genes, there are more than two possible allele options floating around in the population.
More than just, say, A and A.
Precisely. But any one person still only gets two alleles, one from each parent. The ABO blood group is the classic example. There are three main alleles, IA, IB, and IA.
3.
IA tells the body to make A antigens on red blood cells.
Mm-hmm.
ID codes for B antigens, and the I allele. It doesn't code for either antigen.
So A, B, and nothing.
Pretty much. And here's where it gets interesting. IA and ID are codominant with each other.
Ah, like the cattle.
Exactly.
Yeah.
If you inherit both IA and IB, genotype IA-IB, you express both. You have both A and B antigens, so your blood type is AB.
Okay. What about the I allele?
The I allele is recessive to both IA and IB. So if your genotype is IAAA or IAI, your blood type is A.
Because IA dominates I.
Right. Same for B. IBB or IB gives you blood type B. And only if you get two recessive E alleles, genotype I, do you have blood type O with no A or B antigens.
Wow. Okay. So the ABO system combines codominance and simple dominance with multiple alleles. That's a great example. Now let's shift gears slightly. Gregor Mendel, the father of genetics. Why was his work so important?
Mendel. Well, he was an Austrian monk back in the mid-1800s, and he basically figured out the fundamental rules of inheritance using pea plants.
Pea plants, really?
What was revolutionary was his approach. It was systematic, quantitative. He didn't just observe, he counted. He chose pea plants because they had these really clear, distinct, contrasting traits, tall versus short, round versus wrinkled seeds, yellow versus green seeds, stuff like that. 7 traits in total.
And he tracked them carefully.
Meticulously. He crossed specific plants and recorded exactly what traits appeared in the offspring, generation after generation. This allowed him to see patterns nobody else had noticed.
And his famous experiments with the tall and short plants were central to this, weren't they?
Absolutely pivotal. He started with true breeding plants, tall ones that only made tall offspring. Short ones that only made short.
So pure lines.
Exactly. He crossed a true breeding tall, TK, with a true breeding short,
Yeah.
TT. And the first generation, the F1, they were all tall, every single one. Genotype TT.
Even though they had that T allele.
Right. That's dominance in action.
Yeah.
But then, the clever bit, he let these F1 plants self-pollinate. And in the next generation, the F2, boom, the shortness reappeared.
It came back.
It did. And not randomly. He consistently got about 3 tall plants for every 1 short plant. A 3-to-1 ratio. That ratio was the key. It showed inheritance wasn't just blending, it was particular. These discrete factors, alleles, were being passed on.
And from these numbers, he worked out his laws.
Precisely. He formulated 3 fundamental laws. First, the law of segregation. This says that the 2 alleles an individual has for a trait separate or segregate during gamete formation, sperminate.
Yeah.
So each gamete gets only 1 allele.
1 copy per parent.
Yep. Second, the law of dominance.
Yeah.
As we saw in heterozygote TT, the dominant allele T masks the recessive one, determining the phenotype. And third, the law of independent assortment. This says that alleles for different genes sort themselves into gametes independently of each other. So inheriting the allele for tallness doesn't affect whether you inherit the allele for, say, yellow seeds, assuming the genes are on different chromosomes.
So the inheritance of one trait doesn't influence another.
Generally, yes. If the genes aren't linked together on the same chromosome, This independent assortment is crucial for understanding dihybrid crosses, where you track two traits.
And you use diagrams to show this.
Yeah, he developed genetic diagrams, including what we now call Punnett squares. They're tools to visualize crosses and predict the outcomes, the possible genotypes and phenotypes of offspring. Still incredibly useful today.
Absolute foundational stuff. Okay, the source then moves beyond these basics to sex determination and sex-linked inheritance. How does being male or female affect how we inherit traits?
Right, so in humans, sex is determined by our sex chromosomes. Females typically have two X chromosomes XX. Males have one X and one Y XY.
XX female, XY male. Got it.
During meiosis, when eggs and sperm are made, females only produce eggs containing an X chromosome. Males, however, produce sperm that contain either an X or a Y.
50-50 chance for the sperm.
Pretty much. So if an X sperm fertilizes the egg, you get XX, a female. If a Y sperm fertilizes it, you get XY, a male. That's why the chance of having a boy or girl is roughly 50%.
Okay, basic plumbing. But how does this link to inheriting other traits? Sex-linked inheritance.
Well, sex-linked traits are those whose genes are physically located on the sex chromosomes. Usually we're talking about the X chromosome because it's much larger and carried way more genes than the tiny Y chromosome.
So genes on the X or Y?
Mostly the X. And this matters because males only have one X. So whatever allele they get on that single X chromosome, whether it's dominant or recessive, it's going to be expressed. There's no second X chromosome with another allele to potentially mask it.
I see. No backup copy, so to speak.
Exactly. And that's why sex-linked recessive disorders are much more common in males.
Can you give some examples?
Sure. Red-green color blindness and hemophilia are the classic ones. The alleles causing these conditions are recessive and located on the X chromosome.
Okay, color blindness. How does that work?
Let's use symbols. Say XB is the normal vision allele, and XB is the recessive colorblind allele. A female can be XBXB normal, XBXB, normal vision but a carrier, or XBXB, colorblind.
A carrier has the allele but not the condition.
Right, because XB is dominant, but a male.
Yeah.
He's either XBY, normal vision, or XBY, colorblind. If he inherits that one XB, that's it. He's colorblind.
Because the Y doesn't have the vision gene.
Correct. Females need 2 copies of XB to be colorblind, which is much less likely. A carrier female, XBXB, though, can pass the XB allele to her children. Half her sons, on average, would inherit it and be colorblind.
That makes it so much clearer why it affects males more often. The source also mentions hairy pinna on the Y chromosome.
Yeah, that's a rare example of a Y-link trait. Since it's on the Y, only males have it, and it's passed directly from father to son. Females never get it.
Okay. Now, we talked about ABO blood groups earlier as an example of multiple alleles. The source says they can be used in paternity testing. How does that work?
Well, it's more about excluding potential fathers than proving paternity definitively.
How so?
Since we know that genetics type A is IAIA, or IAI, B is IBIB, or AB is IAIB, O is IA, we can look at the mother's, child's, and potential father's blood types. If the potential dad's blood type makes it genetically impossible for him to have contributed the necessary allele to the child, given the mother's contribution, then he can be ruled out.
Can you give an example?
Sure. Say the mother is type O2 and the child is type AB, IAIB. A man with type O2 cannot be the father. Why? Because the child needed an IA allele from one parent and an IB from the other. The mother could only give an I. The type O man also only has I alleles to give. He couldn't have provided the IA or the IB.
So it rules him out completely.
In that case, yes. But crucially, it doesn't prove someone is the father. Other men might have blood types that are compatible. For definitive proof, you really need modern DNA profiling. Blood groups are more of an initial screen or exclusion tool.
Right. DNA is much more specific. Okay, let's circle back to dihybrid crosses. We know they involve 2 traits. How do we actually predict the outcome?
Right. Let's use Mendel's P's again. Say we track stem height, TT, and flower color. PP, P for purple, P for white. Imagine crossing 2 plants that are heterozygous for both. TTPP.
Okay, TTPP crossed with PTPP.
Now, because of independent assortment, the alleles for height, T or T, separate independently from the alleles for color, P or P, when the gametes are formed. So a TTPP plant can make 4 different kinds of gametes, TP, 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 and TP, and it makes them in roughly equal numbers.
4 possible combos from each parent.
Exactly. To figure out the offspring, we use a bigger Punnett square. It needs to be 4 by 4 to show all the possible combinations when these 4 gamete types from one parent meet the 4 from the other. That's 16 squares in total.
16 possibilities, wow.
Yeah. And when you fill it all out and count the resulting phenotypes, assuming independent assortment, you get this classic ratio. 9 displaying both dominant traits, tall purple, 3 displaying one dominant and one recessive, tall white, 3 displaying the other combo, short purple, and 1 displaying both recessive traits, short white.
The 9.3.3.1 ratio.
That's the one. It's the signature of a dihybrid cross between two double heterozygotes when the genes assort independently.
That's a key pattern to remember. Okay, how do we study these patterns in actual human families? The source mentions pedigree diagrams.
Exactly. Pedigrees are basically genetic family trees. They're visual tools used to track a specific trait through multiple generations.
Like a family tree, but with specific symbols for genetics.
Precisely. There are standard symbols, squares for males, circles for females. A horizontal line connecting them means they had children. Vertical lines lead down to the offspring. Affected individuals, those showing the trait, are usually shaded in.
And what can we learn from looking at one?
By analyzing the pattern, who has the trait and who doesn't, and how it passes down, we can often figure out the mode of inheritance. Is it dominant or recessive? Is it possibly sex-linked? We can also often deduce the likely genotypes of individuals in the tree.
So you mentioned earlier traits skipping a generation might suggest it's recessive. Can you elaborate on how we read pedigrees?
Sure. If you see a trait appearing in, say, grandparents and grandchildren, but not the parents in between, that's a strong clue it might be recessive. The parents would likely be carriers, heterozygous, but unaffected.
Okay, and dominant traits.
Dominant traits usually don't skip generations. If a child has it, at least one parent typically has it too. It tends to appear in every generation. Of course, you need to look at the whole picture, check for inconsistencies, but these are good general rules of thumb. Seeing it in every generation strongly points towards dominant inheritance.
That makes sense. It's like detective work using the family tree. Now, the source also covers mutations, these permanent changes in DNA. What kinds are there and what effects can they have?
Right, a mutation is just a permanent change in the DNA sequence. They can happen spontaneously, like errors during DNA copying, or be caused by external factors, mutagens like radiation or certain chemicals.
And are they always bad?
Not necessarily. Their effects vary wildly. Many happen in parts of the DNA that don't code for proteins, so they might be completely harmless, silent. Others, though, can change the protein product, and those can be harmful, leading to genetic disorders, hemophilia, sickle cell anemia, albinism, the color blindness we mentioned.
But sometimes.
Very rarely, a mutation might actually be beneficial. It might give the organism some advantage in its environment, which is key for evolution. But mostly, mutations are neutral or harmful.
The source also mentions gene mutations versus chromosomal aberrations. What's the difference?
Good distinction. Gene mutations are changes within a single gene. Think small scale. Adding, deleting, or swapping one or more DNA bases, like a typo in a word. Chromosomal aberrations are much bigger changes affecting whole chromosomes or large parts of them. This could be a change in the number of chromosomes, like having an extra one, which causes Down syndrome, trisomy 21. Or it could be structural changes, like deletions or duplications of large chromosome segments.
So gene mutation is like a typo. Chromosomal aberration is like messing up whole chapters or pages in the instruction book.
That's a really good analogy, yeah.
Okay.
Captures the difference in scale.
Okay, let's shift now to biotechnology. This is where we apply all this genetic knowledge, right?
Absolutely. Biotechnology uses biological systems or organisms, or parts of them, to create products or processes to, well, improve human life or the environment. The source mentions a few key areas.
Like genetic engineering.
Exactly. Genetic engineering involves directly manipulating an organism's genome, its DNA, to introduce desired changes. This is how we create GMOs, genetically modified organisms, or transgenic organisms, which might contain DNA from a different species.
What are some examples of why we do that?
Oh, loads of applications. In agriculture, we've made crops resistant to pests, so farmers use fewer insecticides. Or crops tolerant to herbicides, making weed control easier. There's even golden rice, engineered to produce vitamin A precursors to help fight deficiency.
And in medicine.
A huge one is using genetically engineered bacteria, or yeast, as tiny factories to produce medicines like human insulin for diabetics, or growth hormone, or vaccines.
The source also brings up stem cells. What's their role in biotech?
Stem cells are fascinating. They're like blank slates, undifferentiated cells that haven't specialized yet. Their superpower is that they can divide and differentiate into many different specialized cell types, muscle, nerve, blood, etc.
Which sounds incredibly useful for medicine.
Immense potential. The idea is to use them to repair or replace damaged tissues. Think treating heart disease by replacing damaged heart muscle, helping burn victims grow new skin, repairing spinal cord injuries, maybe even tackling diseases like Parkinson's or Alzheimer's.
Are there different kinds?
Yeah, broadly, you have embryonic stem cells derived from early embryos, which are pluripotent, can become almost any cell type, but raise ethical debates. And adult stem cells, found in various tissues like bone marrow, which are more limited but still very useful and less controversial. Research is ongoing for both.
And finally, under biotech, cloning. What's that about?
Cloning just means creating a genetically identical copy.
Yeah.
It happens naturally, think identical twins or plants reproducing asexually.
But artificial cloning.
Right, that's what we usually mean in biotech.
Oh.
The famous example is Dolly the sheep back in the 90s. She was created using a technique called somatic cell nuclear transfer.
How did that work, basically?
You take a body cell, somatic cell, from the animal you want to clone. You take its nucleus, containing the DNA, and transfer it into an egg cell from another sheep whose own nucleus has been removed. Then you stimulate that reconstructed egg to develop like an embryo. Dolly was genetically identical to the sheep that donated the body cell nucleus.
So a genetic copy, is it just for making whole animals?
Not necessarily. There's also therapeutic cloning. The goal there isn't to make a whole organism, but to create cloned embryos to harvest their embryonic stem cells. These cells would be genetically matched to a patient, potentially avoiding immune rejection if used for therapies. Still very much in the research phase, though, and also ethically debated.
Okay, so Dolly was reproductive cloning, making an individual. Therapeutic cloning is for making matched stem cells. Got it. Now, the last topic in the source is mitochondrial DNA. What's special about that?
Oh, mtDNA. It's cool because it's not in the cell nucleus with the rest of our chromosomes. It's found in the mitochondria, these cells' energy factories. It's a small, circular piece of DNA.
And why is it interesting for genetics?
Two main reasons. First, we inherit it almost exclusively from our mothers. The mitochondria in the sperm usually don't make it into the egg during fertilization.
So it traces the maternal line.
Exactly. It's passed down from mother to child, mother to child. Second, it tends to mutate at a relatively known and faster rate compared to nuclear DNA.
How is that useful?
These two things make it a fantastic tool for tracing ancestry, especially deep ancestry, through the maternal line. By comparing mtDNA sequences between different people and populations, scientists can figure out how closely related groups are and estimate when their maternal lines diverged.
Does it tell us about human origins?
It's provided major evidence for the out-of-Africa hypothesis, the idea that modern humans originated in Africa and then migrated out to populate the rest of the world. Analyzing global mtDNA patterns strongly supports this. It's like a molecular clock tracing our shared maternal ancestry back through time and geography.
Wow. So mitochondrial DNA gives us this unique window into our deep maternal past.
It really does. A direct line back.
Okay, wow. We have covered a lot of ground today. We've really done a deep dive into genetics, from Mendel's peas and basic inheritance,
Through dominance patterns, sex linkage, those tricky blood groups.
all the way to mutations, pedigrees, and then into modern biotechnology, GMOs, stem cells, cloning.
And finishing with that fascinating look at mitochondrial DNA and our ancestry. It really shows how the field builds from simple principles to incredible complexity and powerful applications.
It absolutely does. It's amazing how those fundamental rules Mendel figured out underpin so much of life. And now, technology.
It's a field that just keeps evolving, constantly giving us new insights into life itself.
Which brings us to our final thought for you, the listener. Now that you've got this deeper understanding of how traits pass down, maybe think about your own family. Are there traits or patterns you see differently now?
Or looking forward, with things like genetic engineering becoming more powerful. What ethical lines do you think are most important for us to consider as we keep unlocking our own biological code?
Lots to think about. We really encourage you to explore anything that sparked your interest today further. Thanks so much for joining us for this deep dive.